Hello. Today I want to talk about microscopic structure of human kidney. When you put a sample of kidney under the microscope, you can see numerous amounts of spherical and tubular shaped structures. These are called as nephrons. Each kidney has about 1 million nephrons. Nephron is the functional unit of the kidney. It means that the main function of the kidney occurs in these structures. Each nephron consists of two distinct parts. Part 1 is a global-shaped structure, which you can see here with number 1. This global-shaped structure itself contains a special kind of capillaries and a surrounding capsule. This capsule is named as Bowman's capsule. So, Bowman's capsule and its inside capillary network together are called as glomerulus. In this slide, you can see two glomerulus. One here and another one here. Both are shown with number 1. The second part of each nephron is a complex tubular structure with different shapes in different locations and also with different ultrastructural anatomy and different physiological functions. Each part of this structure has a special name. In this slide, different parts of this tubular structure are shown by different numbers. Now I want to show them to you. Here, number 2 is called as proximal convoluted tubule. Proximal because it is near to glomerulus and it is the first part of the tubular structure. Convoluted because it is not straight. After that, here we have a third part. It is called as straight part of proximal tubule or pars recta. Pars means part or section. Recta means straight. So both number 2 and number 3 belong to the proximal tubule. After that, we have a U-shaped structure, both here and there. This U-shaped structure is known as loop of Henle. This loop has a descending part. Number 4, here and here. This part is called as thin descending limb. Thin because it is thin, and descending because it descends from outer medulla into the inner parts. Please compare this loop with this one. What are the differences between these two? Think a moment, please. Yes, as you can see, the loop of Henle in this nephron is longer than this one. So we may call this nephron as a long loop nephron. So a long loop nephron is a nephron with a long loop of Henle. On the other hand, we may call this nephron as a short loop nephron because its loop of Henle is shorter compared to the long ones. So this is the first difference between this nephron and this one. Can you tell other differences? Yes, the location of the glomerulus. Please compare the locations of the glomerulus here and here. What is the difference? The glomerulus here is located more superficially. It is in the outer layer of cortex. But here the glomerulus is located in deeper parts of cortex, and it is near to medulla. Here is medulla and here is cortex. Medulla by itself is divided into an outer medulla and inner medulla. And here is papilla. It is the apex of pyramid, about which I have discussed in session 1. So we can call this nephron as a superficial cortical nephron and this one as juxta medullary nephron. Juxta means near to. Juxtamedullary means near to medulla. And another difference. Which part do you have in this nephron but not in this one? Which number is present here and is absent here? Which number? Yes, number 5. Number 5 is called as thin ascending limb, which is not present in short loop nephrons. In short loop nephron, number 4, which is thin descending limb, is connected directly to number 6. But here, in long loop nephron, after number 4, we have number 5 and then number 6. Number 6 is thick ascending limb. As you can see, it is thicker than this part. So this part is thin ascending and this part is thick ascending limb, which you can see in both types of nephrons. Then we reach to number 7. Number 7 is the distal part of thick ascending limb. Again, you can see in this place, this part, in both nephrons. This part is called as macula densa. Macula densa. Macula means spot and densa means consolidated. Macula densa, so, is distal part of thick ascending limb of loop of Henle. It has important physiological roles, which we will talk about them later. As you can see, a part of this segment is colored as a crescent by red. It is like a red crescent. It is because this part of the segment has special type of cells known as juxtaglomerular, known as macula densa cells. So, macula densa cells are located in this part of tubules. Here, as you see, this part of tubule comes very near to the glomerulus. It is very near to glomerulus, and it is located between two branches of vessels. One of these branches is called as afferent arteriole, which brings blood into the glomerulus, and another one is called as efferent arteriole, which drains the blood from the glomerulus. And as it is shown here, the macula densa is located between the afferent and the efferent arterioles. These collection, it means macula densa cells, afferent arteriole and efferent arteriole, is known as juxtaglomerular apparatus. Juxtaglomerular apparatus. Why juxtaglomerular? Because it is near to the glomerulus. As I told you, juxta means near. Juxtaglomerular apparatus also has some major physiological functions. After this part, we have number 8, which is distal tubule. Distal tubule. Number 9, connecting segment. Connecting segment, which connects distal tubule into number 10. Number 10 is collecting duct. Number 10 is collecting duct. As you see, more than one connecting segment is connected to this collecting duct. We have a cortical collecting duct, which is located in the cortex. It is shown by number 10, and a medullary collecting duct, which is placed in the medulla. And this, in turn, can be divided in two different parts. Number 11, outer medullary collecting duct, which is located in the outer medulla, and number 12, inner medullary collecting duct, which is placed in the inner medulla. And finally, we reach to papillae. The urine, which has been formed, is drained from the collecting duct into the papillae, and after that into the minor calyces. Here is the place for minor calyx. This collecting duct can be seen with naked eye without microscope as these stratifications, as these vertical lines in the pyramids. So these vertical lines are collecting ducts which can be seen without microscope. Three main processes of urine formation occurs inside these functional units. It means inside the nephrons. At first, plasma is filtered through this capillary network, and then two other processes occur inside the different parts of tubular structure. Some substances are reabsorbed, it is called reabsorption, and some substances are secreted, it is called secretion. So we have three main processes in urine formation: filtration, reabsorption, and secretion. We'll talk about these processes in more detail later. In summary, we have more than 10 million nephrons in each kidney. The nephron is the main physiological and functional structure of kidney. Three main processes of urine formation, it means filtration, reabsorption, and secretion, occur inside the nephron, and each nephron consists of two main parts. The first part is a spherical structure named as glomerulus, and the second part is tubular structure, which by itself has different segments with different locations, different names, different microscopical structure under the electron microscopy, and also different physiological functions. We will talk about the physiological functions of each part in later sessions. Thank you.